Let's look at the genetic interactions between host and pathogen, and how they come into play part on this. Basically, this part is going to look at what are the genetic interactions between the plants and the pathogens that determine whether or not disease is going to occur. We have the gene for gene theory. This basically states that for every gene in a pathogen that creates virulence, there is a corresponding gene in the host that allows the host to be resistant to that particular pathogen. If we look at things on the general scale, most plants or most hosts are resistant to most pathogens. There are pathogens that come in on specific things, but most things are resistant to most everything else. We do have those things that come in that are specific on specific plants. The gene for gene theory says for every gene for pathogenicity in the pathogen, there is a corresponding gene for resistance in the host. Sometimes those genes for resistance to the host are not expressing themselves. Sometimes they have to be found. Sometimes they have to be monitored to get expression. They become very interesting and very important. When we look at these, we got two different pictures. One is a picture of an elm tree. The elm tree has virtually been wiped out of the United States due to a pathogen that was brought in from outside. We have an exotic pathogen coming in. Our elm trees were never raised with a gene for resistance for that because they have never been exposed to it in their histories. Therefore, they do not have resistance to it. Whenever you have a pathogen coming in, it brings in a new set of genes against a set of genes that are not there. This normally creates devastation. If we look at the other picture, we see a field. This could be plant Y, but you can see that there are varying degrees of growth and varying degrees of susceptibility to a particular pathogen. They're trying to figure out where the genetics are, which things have good genetics, we can use those to breed, which things do not have good genetics, which means that they need to be out. We have terminology that we have to be able to deal with here. These include pathogenicity. Pathogenicity is the ability of an organism to cause disease. Either it's pathogenic or not. Pathogenicity becomes very important. We have what we call virulence. Virulence is the relative capacity of an organism to cause disease. Virulence is relative. You can have something that's pathogenic. That's a yes/no. Virulence, therefore, is on a scale of 1 to 10. How bad is it? We all know people that they get the common cold or they get a hangnail and it seems like they're going to die from it. And other people, the hangnail does absolutely nothing. The virulence problem is in there. Some pathogens are extremely virulent, such as Ebola. Some are not. We have severity. Severity talks about the relative damage to the host that is caused by the pathogen. Sometimes we have severity that is mild. Sometimes severity goes off the deep end and is really bad. We have immunity. Immunity, if we're looking at it, we normally think of it from a human point of view, which is an acquired resistance to an infection that our immune system brings in. Immunity, in this case on plants, really means that the plant is not susceptible to that particular pathogen. When we deal with this, we may have populations that we have to look at to try and find those plants that are immune to see if they can't be used in order to suppress a pathogen. We have escape. Escape says it's the inability of the host to become infected due to a lack of pathogen. If you want to remain free of the common cold during the wintertime, move up to the top of a mountain where you're all by yourself. You will escape the disease because you're not coming in contact with the pathogen. There are certain areas where you can escape pathogens because you're in the middle of the ocean and the pathogen hasn't gotten there yet. All of these are mechanisms that we have of what we call escape. Hypersensitivity says it's an overexpression of severity. We have things that we looked at the shot hole. What is the shot hole? Shot hole's a hypersensitivity. Pathogen comes in, it starts to do something. The plant reacts by going bonkers and killing all the cells around where the pathogen is. And the next thing you know, that part falls out. That's hypersensitivity. Well, hypersensitivity is quite important as well. Here's the question. How can we create disease-resistant plants? This is one of those things that we have to be able to look at. We have what we call specific resistance. Specific resistance is caused by a single gene. We call it vertical resistance. It works real well as long as that's the only gene that we've got. When we look at pathogens, pathogens may have what we call pathotypes or races or varieties. These are genetic segregates of the pathogen. They're still the same organism. They're still the same species. We can look at different types of races and varieties of these pathogens. These become important because when we look at how we're going to suppress things, if you take something like stem rust of wheat, there are over 1,000 known races. These are 1,000 different pathotypes. How are you going to breed against that? If we've got something else, there may only be one pathotype, which means we can breed against it fairly easily. Specific resistance works against a single pathotype, race, or variety. If we're looking at specific resistance, we're only talking about one race. This can be very interesting because it's going to work against that race if that's the only one that we've got. When we talk about this, we often deal with monocyclic diseases. Monocyclic diseases, we've got one chance of infection per year. So if we've got a single gene that works and it works against the only one that's there, we've won. It becomes very important to understand that single gene works against monocyclic type diseases very easily. If we're dealing with polycyclic, now we got a problem. Polycyclic means that we may have a primary inoculum. We've got a primary inoculum for each race. If we knock out the one race, that means that, hey, the other race may come in and may be problematic. This is the problem we've got with the flu virus every year. It works when it works, but when it doesn't work, it really fails. That's because we're trying to pick up one or two races of the pathogen or one or two pathotypes, and we don't get them all. When we talk about polycyclic, we have to have what we call general resistance. General resistance is polygenic, which means there are more than one gene involved. This is going to give you resistance against many different pathotypes or races or varieties. Therefore, this becomes very important as far as genetics goes when you are trying to breed or select an organism for resistance. Is it monocyclic disease? Do we have a polycyclic disease? How many genes do we have involved? The more genes we get involved, the more difficult it's going to be to try and suppress a particular pathogen. This goes back to one of the original stories that we had with stem rust of wheat. What they're doing is they are destroying a barberry plant. Barberry is the alternate host of the stem rust pathogen. If we get rid of the barberry plant, we can't have sexual reproduction in the pathogen, which means we don't get genetic modification. And this is the Farmer's Bulletin from May 1919 that says, destroy the common barberry. E.C. Stakeman was a very prominent pathologist in the United States. He is very well known. He was in charge of the barberry eradication campaign. He shows the proper way to dig them up, get out all the roots, make sure it's dead, it's gone. This becomes a very important part because if we're going to try and eradicate something, we have to make sure that we are not getting any new varieties. You can see here, top one is a turfgrass plot. They are breeding, trying to figure out which variety of a species of turfgrass is going to be susceptible and not susceptible to a specific pathogen. The bottom one looks like some sort of leguminous crop. So on the right-hand side, it's not looking good because we've got something going on. On the left-hand side, that's not looking too bad. They look at yield, they look at production, whatever else is going on. This looks like rice. You can see the part in the foreground is not looking good. The part in the background is looking fine against this particular pathogen. We constantly are trying to find materials that have the ability to resist the pathogen when we look at a genetics program. This is showing you potato. This could be Rhizoctonia. You can see the potato leaves between what they have they're calling resistant, what they're calling medium, and what they are calling susceptible. And you can see a planting in here where the plants in the foreground did not survive. That particular genoplasm is not good for growing in the field when you have got this particular pathogen. So when we look at genetics, genetics becomes very important to understand in the greater scheme of things as far as how do we go out and decrease problems from a specific type of pathogen.